Hello everyone, my name is Benjamin Tebby, and today we'll be talking about the biomedical uses of surface proteins on bacteriophages, gram-positive bacteria, and probiotics. Surface proteins are proteins on the surface of microorganisms that are used for signaling, communication, immunoreactions, and enzymatic activity. So the first topic, bacteriophages. What are phages? Phages are viruses that attack only bacteria. That means that they don't attack human cells, eukaryotic cells. Phages, like other viruses, are a capsule of proteins in which are enclosed genetic information. The phage inserts its genetic information into the host bacterium and takes over the bacteria's replication mechanisms and to produce more phages. The question scientists asked was the manipulation of the surface proteins on phages possible? Phage display technology was the answer to that question. Phage display technology uses two steps. The first is genome modification, and the second is amplification. In genome modification, you begin by creating a phagemid. A phagemid is a gene that codes for a specific protein desired by the scientist. And the second player in this group is a helper phage. A helper phage is just a wild strain of phage with a normal genome. Both are inserted into a host bacterium or to a series of bacteria, and the phagemid competes with the wild type phage for a position on the surface protein gene. Only a few of the phages will express the phagemid gene, and that leads us to the second step. In the second step, the phages are applied to a surface with antigens. So the phages that have been modified, that have fusion proteins on them, will bind to the antigens, and the rest of the phages will be washed away. The bound antigens are eluted and collected and re-injected into a new batch of host bacteria. These bacteria have more helper phages added to them, and the cycle repeats itself. This process is repeated 3 to 4 times until you have a high titer of modified phages displaying the desired protein. A group of scientists use this method, also called biopanning, to develop a phage that will express the human antibody to alpha cobra toxin. The toxin is produced by the monoclid cobra of Southeast Asia and is highly venomous. Scientists were able to identify which antibody binds best to the toxin and are trying to develop an antidote to that toxin with this new discovery. Next thing we'll talk about is gram-positive bacteria. Unlike phages, gram-positive bacteria can attack human cells, and they do so using their surface proteins. For example, Clostridium tetani, the bacteria that causes tetanus, uses its surface proteins to bind to an anaerobic site within the host and before secreting its neurotoxin. Scientists have identified a group of proteins that can be inserted into the surface of gram-positive bacteria. One of these proteins is protein A. Now, protein A displays a motif that other bacteria respond to by attaching to it with a covalent bond. The scientists have been able to modify the opposite end of protein A, the end that is displayed on the surface of the bacteria, and have added different polypeptides, including enzymes and antibodies. A group of scientists were able to add a polypeptide, a protein that binds to metals, specifically nickel and cadmium. Now, these are toxic metals which, if are found in high concentrations in the human blood, in the human system, will cause a lot of damage. So by adding these metal binding proteins to protein A, which was added to the bacteria, scientists have been able to create a sort of antitoxic treatment for organisms such as mice and humans. The last thing we're going to be talking about is probiotics. Probiotics is a catchphrase among health food experts. There's a lot of talk about the benefits of probiotics, and they do have a lot of benefits. Probiotics are a group of bacteria that live in the gut. Lactobacillus and Bifidobacterium are the two main groups of bacteria that comprise of probiotics. Benefits of probiotics include reduced lactose intolerance, resistance to H. pylori infections, and some anti-cancerous activity has been correlated with probiotic presence in the gut of humans. Now, we know that surface proteins on probiotics interact with the gut lining of humans. The exact mechanism by which this happens is still a topic of a lot of debate, but we do know that surface proteins initiate signal responses on the host, which lead to a cascade of signals within the host's system and can result in increased mucose production as well as decreased inflammation, which is helpful if the patient has an inflammatory bowel disease such as colitis. We still have a lot to learn about probiotics, but it is useful to know that surface proteins are where we can start our research. So we looked at phages, we looked at bacteria, we looked at probiotics. All these are different organisms, very different, and they all have different roles in the world around us. But the fascinating thing is that they all can be manipulated and they all can be modified to have beneficial effects in medicine and biomedical research.